the alternating weeks. We'll finish this material, our respiratory physiology section today, as we focus on regional differences and then control of ventilation. So those are the last two main elements in this section before we talk about the spirometry that you'll need next week and the week after. Now I put this slide up last class just to illustrate how effective the lungs are. Question?
Just wondering if you wanted us to come in like any athletic gear, like are we doing any testing?
Okay, for lab today, no testing, no exercise, just basic resting stuff to understand how the equipment works and proper care, things like that. Good question. Every other time you come to lab, you'll need somebody with exercise clothing, and we're going to try to circulate, or not circulate, but alternate the individuals that are exercising and the roles that you have within your group so everyone gets a chance doing something different. Okay? So this slide just illustrated how effective the lungs are normally at exchanging oxygen. And we talked about a whole list of reasons why the lungs are ideally suited to exchange gases according to the Fick law of diffusion. But this is average. So while we have a depiction here of transit time in a capillary, not every capillary follows this same response. This is the net sum of all of the capillaries in the lung, ideally, that have quick onloading of oxygen within a third of the transit time under normal conditions. So the factors that affect how likely gas is to be exchanged are what we discussed at the end of last class, the ventilation at the individual alveolus and the perfusion of that alveolus in the capillaries. So the ventilation-perfusion ratio will move this relationship. We'll flatten it, we'll accelerate it, and we're going to talk about the different locations in the lung where ventilation-perfusion ratio varies. One way to start our discussion of this is to consider how the abnormal trace might be produced. How might the abnormal trace be produced by a change in either ventilation and or perfusion? If we want, ideally, there to be just enough air to fully saturate the blood that's available, how might that situation change in the abnormal case?
There's too many red blood cells, the capillaries could be thicker, or like the alveoli membrane could be a little bit thicker, so perfusion wouldn't
Okay.
easily.
Okay, I like your first point, and the second point is true that the capillaries could be thicker, but let's assume the mechanics of the lung are consistent. But your first point is well taken, that there might be too many red blood cells. Or the flip side is there's not enough air. Ventilation is lower and or perfusion is higher. So the ratio would be 1? No? How does it change? Less than 1. Right, the ventilation-perfusion ratio is less than 1. It's less than ideal. Typically, that kind of mismatch will result in this abnormal trace, which we think of as a type 1 respiratory failure. It's not often that it will progress to this grossly abnormal trace, which we consider a type 2 respiratory failure. And I'll just briefly outline what I mean by that on this slide, which is a new addition to the slides that are on Moodle. It's a brief overview of these two types of respiratory failures. So type 1 is a situation where you might be at altitude, something that results in a small ventilation-perfusion mismatch. It could be an embolism, so a blockage in the pulmonary vasculature. All of these things will typically reduce PO2, but PCO2 is normal-ish, around 40, but it's under 50, which is a threshold that we consider when discussing respiratory failure. So type 1 respiratory failure characterized by hypoxemia, but normal PCO2. Small disruptions in the ventilation-perfusion ratio result in this type of respiratory failure. Whereas the more severe type still exhibits hypoxemia, which is our word for a low PO2, and hypercapnia is the main difference, so a high PCO2 over this arbitrary threshold of 50 millimeters of mercury. So more extreme variations in the ventilation-perfusion ratio caused by either airway resistance, so chronic obstructive pulmonary disease is a big one. We'll talk about that at length next week. Or a change in the mechanics of the lung, reduction in surface area from bronchitis or a thickening of the walls. All of those are characteristics of COPD. Medications, extreme obesity can influence the mechanics and the structure of the lung and compromise the ventilation-perfusion ratio as well. So these are the more clinical types of disturbance that we'll talk about next week. Normal variations in ventilation-perfusion might cause this these type 1 characteristics in regions of the lung, in individual regions. So how might we go about measuring ventilation and perfusion across the lung? Really, how will we go about measuring ventilation? Perfusion is a bit more involved. But we have an idea that ventilation changes as you move from the apex or the top of the lung to the base through some classic studies that had the subject inhale this stable xenon isotope. And xenon will radiate through tissues easily. And so if you set up a detector in line with the person or in line with the vertical aspect of the lungs, you can see how many of those molecules or atoms are transmitted out of the lungs at varying levels. And the more atoms you have transmitted out, generally higher the ventilation. The fewer the atoms are transmitted out or detected, the lower the ventilation. And importantly, before we talk about how ventilation changes, I think it's worthwhile noting that most of the differences are due to gravity. So think about the lungs almost as if they're hanging from the trachea within the pleural cavity. And they are. They're not supported by much else. They're typically hanging by the trachea here, and the effects of gravity will affect the lung differently at the apex, the top of the lung, or the base. There might be more of a pull at the apex and perhaps more of a crowding or a compacting of the lung tissue at the base if it butts up against the diaphragm. Now, what this will result in is a deformation of the alveoli. So at the top, the alveoli tend to be larger, they're stretched. And at the base, the alveoli tend to be a bit more compacted. At first glance, this seems as though, okay, well, I'd imagine ventilation to be lower at the base. If the alveoli are smaller, less air can reach them, so ventilation should be lower. Therefore, ventilation should be higher at the top if they are larger alveoli. But that's not, it's counterintuitive because it seems that should be the case based on the initial size of the alveoli, but what we're concerned about is the change with inspiration. When the respiratory muscles contract to inflate the lungs, the alveoli at the apex don't have a lot more room to expand. They're already somewhat expanded. They might be tense or stiff, not rigid, but the walls tend to be stiffer, and there isn't much more room for them to stretch. Whereas at the base, starting somewhat compacted, smaller, contained, they're what we call more compliant. They have the ability to expand much beyond their original size. And it's the expansion that dictates ventilation. It's the expansion that creates negative pressure in the alveoli. And that negative pressure creates a gradient for air to flow from the outside to the inside of the lungs. And the larger that gradient, the more airflow you have. So the alveoli at the base of the lung, with their increased ability to expand, we'll see more ventilation. We'll see more airflow. And that's exactly what we register, we detect when we monitor the changes in the emittance of this xenon isotope. Fewer atoms at the top, more at the bottom, indicating higher ventilation at the base of the lung. So perfusion stayed the same. The ventilation-perfusion ratio would go up as you reach the base of the lung. It would go down towards the top. Make sense? Largely due to the effects of gravity and the way the lungs are structured within the thoracic cavity. I'll also point out in this discussion when I'm referring to ventilation, I mean alveolar ventilation or the usable ventilation. This is slightly different than whole body ventilation that we think of or that we record with the metabolic cart, VE, minute ventilation. The difference between the two is that minute ventilation includes both alveolar ventilation and air that goes to the dead space in the lungs, the trachea, the conducting zone, parts of the lung that aren't involved in gas exchange. Alveolar ventilation, only that portion of volume that is involved in gas exchange, that reaches the respiratory zone. So greater alveolar ventilation near the base of the lung. How about perfusion? You get to see the trace early. Perfusion, similar to ventilation, is higher near the base of the lung. Again, the effects here are largely due to gravity, not so much the distension of the capillaries, although there is a small effect of the shape and changing the conformation of the capillaries. More so here, the position of the capillaries in relation to the level of the heart. Remember, pressure in the respiratory vasculature is low, very low. If we think the arterial systemic side of the heart, we're thinking 120 millimeters of mercury during systole, 80 during diastole. Those are high. Pulmonary circulation might exhibit pressures in the 5 millimeters of mercury range, very, very low. And so gravity here has a large role to play. Mainly, water flows downhill. Blood will more likely accumulate under the level of the heart than above, even though some does perfuse the apex of the lung. So largely due to gravity. The compaction of the lung at the base also does help flow. We call this transmural pressure. So the likelihood that blood will be pushed through the capillaries is greater at the base than it is at the apex. The effect of gravity compacting the lungs more near the base will help push blood through. There's less of that effect happening at the apex of the lungs. So those two factors contribute to a higher perfusion at the base of the lung than at the apex. Yeah?
What did you call that? Give it a name.
Hydrostatic pressure.
Transmural?
Transmural or hydrostatic pressure, yeah. Yeah, transmural across the membranes between the pleural cavity and the capillaries, and hydrostatic is another word for the pressure within the capillaries. Yeah, good clarification.
So does that mean that the pressure in the capillaries will be more or less?
It will be influenced by the compaction more, so it will be higher, and I'll go into it in a second as we get to the inset figures at the top and the bottom, but it will be higher, pushing blood through the capillary network more. So that is coming up right now. Okay, so these inset figures describe that relationship. Describe the relationship of pressures, and there's three types of relationships. There's three possible outcomes called Zone 1, Zone 2, and Zone 3, and the only physiological outcomes are Zone 2 and Zone 3. Zone 2 is at the top, Zone 3 is at the bottom. And these inset figures relate the pressures relatively between the arterial side, the alveolus, and the venous side of the capillary. Remember the arterial system in the pulmonary circulation supplies deoxygenated blood. This is incoming to the capillary. Gas exchange occurs, and the pulmonary vein supplies oxygenated blood back to the heart. So blood flow here is moving from the left to the right, and in this situation, zone 2 at the top, the arterial pressure is higher than the alveolar pressure, which is higher than the venous pressure. This describes a situation where with every heartbeat during systole, the capillary opens up. During systole, this happens, where the arterial pressure briefly rises above that of the alveolus. Pressure gradient dictates that blood will flow through then to the vein. And this is characteristic of the alveoli and capillary networks at the top of the lung, at the apex of the lung. They're typically closed or flow is impaired during diastole, and then with every heartbeat, blood is pushed through. So flow is intermittent. Flow is impeded. It's not always flowing. Whereas at the base of the lung, we observe what we call zone 3 respiration, where the arterial pressure is larger than the venous pressure, and the lowest pressure is found in the alveolus. This describes a situation where there is always flow between the arterial side and the venous side of the capillaries, because the alveolar pressure never impedes the flow in the capillaries. So you can almost think of these pictures as the large circle being the alveolus and pressing down on the capillary, and the swaying or the movement, the bulging of the artery is a bit over-exaggerated. It's closed off at the top. It's opened. It can't be opened more at the base unless there's a negative pressure, which doesn't often happen. But it's open all the time at the base, allowing flow through the capillary. The alveolus itself doesn't impede that flow as much. The zone 1 that we haven't discussed is just the last remaining configuration, where alveolar pressure is higher than any capillary pressure, and flow is blocked always. That doesn't happen in practice, so it's not discussed here at length. Okay? Zone 1 is where alveolar pressure is higher than vascular pressure, and so there is no flow, but that doesn't occur physiologically. Zone 2 at the apex, zone 3 at the base, intermittent flow at the apex, constant flow at the base. Make sense? With repetition and review, it'll make more sense. So what then is the result as far as the ventilation-perfusion ratio? Both of these are lower near the top and higher at the base. Ventilation and perfusion are both lower at the top, higher near the base. You can see the numbers briefly pop up here on the right, but I think it's easier to show you on the next slide how they are related. So we have ventilation, again, that's higher at the base, lower at the top. So there's a declining relationship where ventilation is reduced as you move towards the apex of the lung. Here the apex versus the base of the lung is simply represented by the rib number, the horizontal slice or section where you would find that part of the lung in the body. And blood flow exhibits the same relationship. Higher at the base, lower at the top, but the relationship is steeper, such that the change as you move vertically is greater than the change in ventilation. If these were, if they had the same slope, the same consistent slope, ventilation perfusion would always be 1. They would always be matched. They would just be both higher or both lower, depending on where in the lung you looked. But blood flow is higher at the base than ventilation, and the opposite is true at the top, at the apex. Blood flow is lower at the top of the lung than ventilation is. So those effects of gravity that we talked about seem to have a larger relative effect on blood flow than they do on ventilation. So when we're in a situation, like at the base of the lung, where flow is greater than ventilation, the ventilation-perfusion ratio is less than 1. Smaller number in the numerator, larger number in the denominator of the ratio gives us a fraction less than 1. At some point in the lung, those cross over and are equal. After that point, as we progress superior, or to the apex of the lung, a higher ventilation and a lower flow means the relationship is larger than 1. Higher number in the numerator, lower number in the denominator. 2 divided by 1 is more than 1, it's 2. So how does this relate to the overall ventilation perfusion matching in the lung? All of these sections exhibit various ratios across a spectrum from maybe 0.5 to 7 at the base, up to 3 at the apex. When we describe the overall ventilation perfusion ratio, it's the average of all of these values. It ends up being that the ventilation perfusion ratio is somewhere close to 0.8 for the lung as a whole. Somewhat less than perfect for the lung as a whole. This same information is shown in a slightly different way here, translating how that ratio affects the gases within the blood. Question at the back.
Question. So whenever it said like it can go up to 3, does that mean the ventilation perfusion ratio can be 3?
The ventilation perfusion ratio can be 3. That is, ventilation is relatively 3 times higher than perfusion. Yeah.
But it typically doesn't go higher than that, or?
I would say it typically doesn't go higher than that. No, and that's in the extreme at the apex. Yeah. But relatively, that's a small influence on the overall ventilation perfusion ratio. Remember, all of those capillaries drain into one central pool as they return to the lung. So the deoxygenated blood from the apex and the oxygenated blood from the base will combine to result in that characteristic 100 millimeters of mercury PO2 and 40 millimeters of mercury, PCO2 that we see in the cascade slide overall. So that is essentially shown here, how the varying ventilation perfusion ratios affect local PO2 and PCO2, and then how all those local concentrations combine to result in the overall PO2 and overall PCO2 that we see from the lung as a whole, as an organ. Again, decreasing ventilation perfusion ratio near the base, and the resulting effect on PO2 and PCO2, and the complementary increase and the resulting PO2 and PCO2 at the apex. The next sheet is not new information, but I think this is a nice sort of cheat sheet. I have a couple cheat sheets to show you. To think about the lung physically as it's oriented within the body, here's one lobe of the lung. We're contrasting the changes due to gravity and ventilation on the left and perfusion on the right. This might help organize your thoughts and ideas a bit more as you review to understand how the ratio is affected if it's not immediately clear. And then on the slide after this one, I have just a brief table that categorizes with respect to some main markers or main thresholds how those zones of the lungs change. So in theory, any mismatch will decrease PO2 and will act to increase the retention of CO2, but it's not often under regular normal circumstances where you will observe hypercapnia, the retention of CO2 in the arterial system. Usually chronic disease and other complications will cause that to occur. Yeah, so when there is a mismatch, it's normally PO2 that's decreased. Arterial CO2 is not affected as much. Those locations where the ventilation-perfusion mismatch is larger than 1 is called wasted ventilation. There's more air available or delivered to those capillary beds than is needed. We don't like wasting energy like that in the body. We try to be very economical, and wasted ventilation or dead space is not something that the body typically likes to allow. The opposite did what I say make sense? I think so. Extra ventilation required above normal is called wasted ventilation. So if you need to increase ventilation above what is being delivered, I think I said it in reverse. If you need to increase ventilation above what is being delivered, so if there is a low ventilation-perfusion ratio, it's called wasted ventilation. Lung units with a high ventilation-perfusion ratio that are inefficient at eliminating CO2 are called dead space. So in terms of the general zones being average, upper, and lower parts of the lung, we would define them or categorize them as such, where the average ventilation-perfusion ratio overall is roughly 0.8, and that is the line in the sand that we draw between the apex and the base of the lung. And the main factors, the main effects of changing that ventilation-perfusion ratio are in affecting PO2, but in some cases PCO2 will be affected as well in extreme chronic conditions. When we're near the base of the lung and the ventilation-perfusion ratio is lower, ventilation is lower than perfusion, we have inadequate gas exchange, inadequate offloading of CO2, inadequate loading of O2, and a resulting decrease in pH, which will become important as we talk about the regulation of ventilation. A resulting decrease in pH. So if the lung overall were to exhibit an average ventilation-perfusion ratio less than 0.8, this would be a concern, this decrease in pH or the increase in acidity. While this does happen in regions of the lung, at the base of the lung, on average, we usually live in this middle column here, the middle of the lung or the lung as a whole, where the ventilation-perfusion ratio is 0.8, and we have full gas exchange, no difference in pH. We're at a neutral 7.7. At the apex, where we identified ventilation is higher than perfusion, it's possible that gas exchange progresses to more than the ideal. We have a greater movement of oxygen across the membrane, a greater movement of CO2, resulting in a higher PO2 in that blood from the capillaries. And this should be a lower PCO2. That is a typo. Less than 40 millimeters of mercury PCO2. Overall, lower PCO2 and a resulting alkalosis, an increase in pH. Again, not common that that happens in the lung overall as an organ. There are regions within the lung that exhibit this, but on average we live in this middle column here. When we talk about chronic conditions like COPD, that might change. We might deviate towards one side or the other. What we haven't talked about is how ventilation and or perfusion can be modified locally. There are actually regions within the lung where the capillaries, if they detect hypoxemia, will constrict. And this is a unique reflex in lung tissue, so that if there's inadequate ventilation, the body will shut down delivery of blood to that region. If there's inadequate ventilation, if gas exchange is incomplete, and the blood there locally is hypoxic, the vasculature constricts to avoid sending blood to that region. It's not going to be able to do its job in that region. I don't think are going to concern ourselves so much with the local reflex activity. That's an example of local control. What is interesting is how this changes with exercise, or how it's corrected with exercise. Largely the effect of exercise is due to the increased activation of the respiratory muscles. And that simply acts to increase the negative pressure drawing air and blood into the alveoli and the capillaries respectively. So we have increases in the suction effect of breathing that help to facilitate movement. Here these are capillaries, these are alveoli shown on the left. And if there are ever situations where some alveoli are closed, as are shown in the middle here on the left, we can correct that through exercise by increasing the recruitment of new channels, so opening up new channels by increasing the suction effect of breathing, or by increasing the distension of those channels. This is largely due to the increase in pulmonary pressure in the vasculature, helping to open up those new channels. If there are mechanical issues where the blockage is not due to over or under inflation, if there is an obstruction or fibrosis, this is less likely to occur. But in those alveoli that are supple or compliant, this is how we can increase ventilation and better match perfusion during exercise. So if there's a mismatch in the ventilation-perfusion ratio, we have inadequate loading of O2, inadequate disposal of CO2, and therefore oxygen delivery might be limited to the rest of the body. Typically, in a normal, healthy individual, that's not a concern. We'll see next week where that might be a concern for individuals with chronic disease. Lastly, on our slide, we talked about this briefly last class, the HbO2, or hemoglobin oxygen efficiency. While this changes with temperature and pH, it usually varies within a consistent range. And so complications here do not generally result in a limitation to oxygen delivery, except where there's some genetic defect in hemoglobin. So let's carry the idea of altering pH and the regulation of ventilation. This should also be a review from A&P. But the factors that affect the reflex to breathe are largely due to changes in pH. Changes in pH are very intimately linked to changes in CO2, or the PCO2 of arterial blood. So we have this sliding scale, if you will, where on average we live here at this black dot. And if the apex and the base with their different characteristic PO2 and PCO2s combine to give us what we expect normally in the arterial blood, we'll stay at this black dot. If there is some disturbance where the overall ventilation-perfusion ratio changes, PO2 and the PCO2 will change accordingly. So if we move to the right with an increasing ventilation-perfusion ratio, we see higher PO2 and lower PCO2. And the reverse is true. If the overall ventilation-perfusion ratio of the lung decreases, we see lower PO2 and somewhat higher PCO2. We identify that changes in PCO2 are not very likely, and this illustrates why those changes are small if they exist at all. But luckily, the drive to breathe is very sensitive to changes specifically in PCO2. This is a trace showing you the ventilatory response, here whole body minute ventilation, not alveolar ventilation, in response to varying arterial PCO2. Now ignore the two lines towards the left, the top and the left, focused on the one on the right just at the start, the open circles. This is the normal situation. Normally, if we have an alveolar PCO2, say around 40, and a minute ventilation of 12 to 15, those values check out with what we identified as a normal resting ventilation and what the oxygen cascades always tell us. If we see even a minute increase in PCO2, ventilation spikes. There are disproportionately large increases in ventilation for small changes in PCO2. This type of response is not characteristic of changes in PO2, which is maybe a bit counterintuitive because oxygen is the thing that we need to live. It's the endpoint where we donate electrons in the electron transport chain. We use that to help rip electrons from food to satisfy our energetic processes. If we're without oxygen, we suffocate. You'd think that the response would be as sensitive to PO2, but that's not the case. PCO2 is by far more important from the body's point of view in regulating ventilation. That's not to say that PO2 has no effect, and this is where the other lines come into play. In a normal situation on the right, with our resting PCO2 and ventilation, that's assuming a PO2 of around 100 or just over 100 millimeters of mercury. 169, I think, is kPa or kilopascals. So that's a normal situation. If we were ever to enter a hypoxic situation where the PO2 dropped here to 47 in the middle line, in the middle example, that same PCO2 now has almost double, it's almost off the charts, a rate of ventilation. A lower PCO2 and a smaller change will cause a much larger spike in ventilation at a lower PO2. So PO2 will grade the body's response to a change in PCO2. If PO2 is lower, the body becomes more sensitive to changes in PCO2. And in an extreme situation where PO2 falls here to an extremely low 37 millimeters of mercury, which is very unlikely, resting ventilation never gets below 20 liters per minute. And in most situations at rest, you'll be sitting towards 50 or 60 liters per minute, hyperventilating, trying to expel CO2. So the body is more responsive to changes in PCO2, but that response is graded based on the PO2 in the alveoli. Why are we more sensitive to changes in PCO2? It all has to do with the relationship between CO2 and the acid-base balance in the body. And this is an equation that you've seen in biology and biochem and anatomy and physiology. It should be fairly common, or at least somewhat familiar. This is the equation relating an abundance or a change in CO2 in the blood. Usually we refer to this in the blood through the actions of carbonic anhydrase, which is the superscript Ca here, and how it can produce bicarbonate and free hydrogen ions. This equation is what we call a near equilibrium equation. You add substrates and you make products automatically. You take away substrates and those products are moved back towards the left of this equation to resynthesize or to replace that lack of substrates. So it's like a seesaw. If there's ever an overabundance on one side or the other, this equation will move so that it is in equilibrium. If we add CO2 to this equation, to this situation, we very quickly then will dissociate. It'll combine with water, make H2CO3, which dissociates into bicarbonate and a proton. If we ever increase the CO2 content, then we very quickly increase the hydrogen ion content. And by definition, the acidity of the surrounding tissue. Right? pH is a measure of the concentration of hydrogen ions. If that concentration goes up, pH goes down, acidity goes up. So there's a very intimate link here between CO2 in solution and then the acidity of that solution. We want to regulate body pH about a neutral range, and if this occurs readily and easily, a small increase in CO2 will cause a small increase in acidity. A large increase in CO2 will cause a large increase in acidity. And it's typically incompatible with the function of enzymes, the structure of cells, and life as a whole if that acidity is too large, varies too much away from the normal neutral range. So when is CO2 produced in the body? We typically see it as a result of cellular respiration. CO2 is produced in active cells within the body. This is a generic active cell and needs to be transported to the lungs for dissipation to the environment. We can typically do that in three ways. One is to dissolve CO2 in plasma. Like oxygen, CO2 does not readily dissolve in plasma, although relatively it does dissolve a bit more. That constant, the dissolving constant, is higher for CO2 than it is for O2, so relatively more will dissolve in plasma than will oxygen, but this is still a very small portion of the transport. The second type is that we can bind CO2 with proteins within the hemoglobin complex, forming what we call carbaminohemoglobin, indicating that it binds specifically to amino groups within the hemoglobin. And then the third and the largest typical form of this transport is through the reaction that we just discussed. CO2 combines with water, forming H2CO3 that dissociates into bicarb and free protons. Typically, the relative contributions are about 10% for vehicle 1, 30% for vehicle 2, and 60% for vehicle or method 3. So the majority travels as bicarbonate in the bloodstream. That means the majority of cellular CO2 that is produced will create a change or affect the acidity of the blood. As this circulates through the body, that carries that change with it through to other tissues as well. Now normally we counteract this by, at the lung, disposing of that CO2, that metabolically produced CO2. We have venous blood coming in, or arterial blood from the pulmonary artery, but deoxygenated blood coming into the alveolus, where all three of those CO2 transport methods are reversed. So CO2 comes out of solution, CO2 disengages from the hemoglobin molecule, and it also flows backwards through the carbonic anhydrase reaction because of the pull, a lower CO2 within the alveolus to remove CO2 from solution. So it makes sense, if we are responsive to CO2, to increase ventilation in order to prevent the accumulation of protons. Now, one common catching point here is that, well, if you combine CO2 and water and H2CO3 dissociates into bicarbonate and a proton, the charges equalize. Bicarbonate has a negative charge, the hydrogen ion has a positive charge, so there's no net difference in charge. And that's true, but we don't care so much about the net charge. We only care about the concentration of protons. That's the definition of acidity. That's what pH measures. It's the change in the concentration of protons. It's not the charge, the net charge difference. It is the concentration of protons in solution. Now, this is largely sensed by our old friends, the central chemoreceptors. The drive to breathe doesn't come from the lung. The drive to breathe doesn't come from the tissues. The drive to breathe comes from specialized cell bodies in very specific locations that we call chemoreceptors. The central chemoreceptors are located in the medulla of the brain, which is a very special area. Not all of it, but a large part of the medulla is not enclosed by the blood-brain barrier, and so blood freely washes over it so it can sense constituent changes in the blood, ions, temperature even. And this regulates bodily processes like sweating, respiration in this case, digestion, a whole lot of other factors. Even in the areas where there is a blood-brain barrier, and I'm indicating that here with this thickened blackened line, CO2 readily passes through. Many other proteins and complexes do not. Typically, if it's a polar molecule, or if it's not lipid soluble, it's contained within the bloodstream. Steroids can cross, CO2 can cross, thankfully caffeine can cross. Very few substances get across the blood-brain barrier, CO2 being one of them, diffuses into the cerebrospinal fluid where, again, carbonic anhydrase is present, and this reaction takes place. If we have a delivery of CO2, a net abundance of CO2, the reaction in this case, as it's written here, proceeds to the left, and this increase in acidity is detected by the central chemoreceptor in the medulla. This specifically sends the signal to breathe. And it is the increase in acidity as a result of the increase in PCO2. It's not PCO2 directly, which I'll show you coming up, which is interesting. This is where the main drive to breathe comes from, and the central chemoreceptors are primarily receptive to changes in PCO2 and hydrogen ions. There are peripheral chemoreceptors that are less responsive to CO2, more sensitive to changes in PO2 that modulate the drive to breathe. And modulate, if I were to put a number on it, to the tune of 20% of the drive to breathe. 80% comes from the central chemoreceptors and the change in PCO2 and resulting acidity. 20% of that drive might come from the peripheral chemoreceptors and changes in PO2. Notice that this stylized figure on the bottom left is showing you just percent of the maximal signal, and when we register a physiological or meaningful change. For normal arterial PO2s, no real stimulus or modulation of the drive to breathe. Even for compromised PO2s, say around 80 millimeters of mercury, there's little signal being sent to the respiratory centers in the brain. These really only kick in as a last line of defense when arterial PO2 falls very, very low to stimulate the drive to breathe here in an attempt to onload O2. But overall, this is a relatively small contributor to the drive to breathe. So questions that you might have left are, well, if these chemoreceptors are to sense a change in the blood and initiate a change in ventilation, what might be that driving force? Is it arterial pH? Does that change with exercise? Is it CO2? Does that change as we move to exercise or in other chronic conditions? O2? And our initial perception is that these factors don't change much with exercise. During really intense exercise, you might register a change in pH. But overall, PO2 and PCO2 are still pretty constant, even during really intense exercise. The lungs are very good at exchanging gas between the blood and the environment. So maybe we have a hint that pH is what is our driving factor. But if we're waiting for changes in PO2 and PCO2 to initiate the response to breathe, we'll be waiting a long time. It's also interesting to consider this because it implies that there is some higher order practiced response. We've learned this as we've developed, perhaps, or as we've engaged in regular exercise. When we make the voluntary choice to exercise, maybe that initiates the response to breathe before and in advance of changes in O2 and CO2. It would be catastrophic to see large variations in PCO2 within the body, so there might be some feed-forward mechanism in the brain that increases ventilation before that signal can respond. If we take a look within the body, it seems that pH is probably the largest driver of ventilation, not so much O2 and CO2. And these three columns are exercise at different intensities, and this is reported in an odd way. So on the left we have 80% of lactate threshold, not 80% of VO2 max. 80% of lactate threshold, and I suppose this is the way that you would want to report it if you are concerned about acidity and PO2 and PCO2 and not so much concerned about the relative intensity of exercise. Lactate threshold is also the threshold where pH changes accumulate. So 80% of lactate threshold is an easy, light workload where we don't register changes in lactate. We don't register changes in arterial CO2 or O2 for that matter, but O2 is not on here. And pH doesn't change appreciably either. The next two columns are describing how far from lactate threshold to VO2 max you're going. This is 35% of delta, which is 35% of the way between lactate threshold and VO2 max. So we'd call that a moderate to high workload. And then 75% of delta, which is 75% of the way from lactate threshold to VO2 max. If you were to ballpark a number, I might say that 35% of delta is 70 or 75% of VO2 max, and 75% of delta on the right might be 90 to 95% of VO2 max, maybe 90. This is where we get to see the factors that change and are more likely to increase ventilation. So we see the accumulation of lactate, like we would expect, being above the lactate threshold. This is the indicator that acidity is being produced at the muscle. And that acidity we see culminate in a decrease in pH here, but we see no appreciable change in arterial CO2 contents. Even though our classic understanding is that CO2 drives ventilation, according to that first graph that I showed you, the appearance of CO2 doesn't seem to be related to an increase in ventilation because it's not changing very much. We don't register large changes in arterial CO2 even at these high workloads. What we do see respond to exercise is pH, the drop in pH. And that implies that this drop in pH is probably the signal that is sensed by the central chemoreceptors to initiate the change in ventilation. And it goes without saying, of course, ventilation is increased with exercise and progressively more as you proceed towards the right of the slide. So probably changes in pH because we don't register changes in CO2. So let's summarize. We've covered a lot today, a lot of complicated topics. I've seen some furious note-taking. Let's summarize. This section, we've identified the lung as ideally suited for gas exchange. According to the Fick law of diffusion, the high surface area, the low wall thickness, and the pressure differential make it suited for gas exchange. Fick law of diffusion is shown here, being proportional to area, pressure, and inversely proportional to the thickness of the membrane. Today we identified the lung has regional differences with respect to how gas moves across the membrane. The bottom of the lung, you might say, is more efficient. It has a higher ventilation and a higher flow, a higher perfusion, whereas the top of the lung is perhaps less efficient. There's often more air available than blood to be saturated. During exercise, we can modify ventilation and perfusion through distension and recruitment of new alveoli. I think another important summary point between this one and the last one would be the effect of gravity on ventilation and perfusion. So we identified the effect of gravity on the alveoli is to compact those at the base, which allows them to expand more and receive more air. And the effect of gravity on perfusion, one, blood flowing downhill below the level of the heart will aid perfusion at the base of the lung. Two, the compaction of lung tissue at the base will increase that hydrostatic or transmural pressure that helps to push blood through the capillaries, again aiding in perfusion. We just talked about how respiration or ventilation is controlled mostly by central chemoreceptors, and what I alluded to is this idea of central command, making the conscious choice to exercise probably also invokes neurons or pathways that increase respiration. And this response is, of course, modified and augmented by the chemoreceptors, the central being responsive to changes in pH, mostly. You can also say PCO2 because it's almost a one-to-one relationship. If you have an increase in CO2, you'll see an increase in the acidity or a decrease in pH. And the peripheral chemoreceptors respond to a decrease in oxygen, but only in situations where it is critical. Both of them respond to arterial blood, and again, pH is likely the main driving factor stimulating ventilation initially.